This is a demonstration of Trevigen's Comet Analysis Software. The software is designed to analyze digital images of fields of cells which have been run in the COMET assay, and then to quantify the degree of damage of each cell. So once the COMET Analysis software has been installed on your computer, a COMET application icon is automatically created on the desktop. So let's click on the icon to begin. The first step in using the new software, you need to begin by creating a new study. So for this demonstration, we'll go under File and we'll hit New Study. So we're going to give a study name. For this study, we're going to call it 4X CometChip Test Images. And my study used 3 slides. And for my slide type, they were 2 well. And to begin with, let's use a default protocol called 4X CometChip alkaline comet. And then we're going to hit Save. So the Comet wizard comes up, and he's very helpful trying to navigate the software. And what you want to note initially is what we're doing here is in that first step, we are creating a special folder, which we called the study name 4X CometChip Test Images. And this is where we're going to store our images so that the software can analyze it. The wizard is asking, do you want to add images now using the Well Explorer? So let's select yes. So in this step, this is the Well Explorer window. We have our study name, 4X CometChip Test Images. We have a protocol name, which we selected, 4X CometChip alkaline comet. This protocol is what we're going to use to identify the scoreable comets that are within your image. And you always have the ability to change this protocol. So we have a Change Protocol button here. All right, so let's go get our images, and we are going to drag and drop our images into the slide wells. So let's use Well Explorer. I have a set of images here. So in this window, you can see that you have slide numbers 1, 2, and 3. And since we did 2 wells, we have position 1 and 2. So it's very easy. All you're going to do is take your image, and you're just going to drop them in there. Then you will move on to your second slide. You're just dragging and dropping. So the important things to note are you can actually drag and drop multiple slides into a well depending on how many pictures you actually took. The type of image files that the software uses are TIFF, JPEG, or PNGs. Some other useful information in this window is you can actually highlight the slide. If you made a mistake dragging the image into the well, you can always delete the file. And if you'd actually like to see what the picture looks like, you can actually open the file. You also have the option that if you don't want to drag and drop, which I actually think is the most useful, you can always browse for your individual images in order to load them into the well. All right, so now let's select Done. So the third step is the wizard now is asking, do you want to open the scanning activity? So this is the third Comet task. So you see step 1, you can always move between steps, but we created the study. Step 2, we added the images using the Well Explorer. And now the third step is, do you want to scan the images? So let's select yes. And now we are in a new window pops up. This is called Scan Mode. Oh, one thing you should note, at the top of the software program, you actually know what study or experiment you're in. It's listed at the top here. Okay, so we haven't actually started to scan yet. But what you can see here is that, again, you have your study name. This is the protocol we're going to use during the scanning. Again, we had 3 slides, 2 wells per slide. In this window, you can see that our scan has not started yet and the software hasn't found any comets. So let's go ahead and say to start the scan. So each of our TIFF images are being brought up, and you're seeing these white boxes which are identifying all of the scoreable comets within that image. So the images I actually chose were based off of CometChip technology. These were 4X images that were actually taken. So the fourth step we're getting ready to go into, the Comet Wizard has now opened up, asking, do you want to review and edit the study now? So let's select yes. So 3 windows now automatically open up. There is also, as you noticed in the previous, when we were scanning, the white boxes were being opened up. We'd like to open up those windows also. So go under Windows and hit Activate Search Images. So if you have multiple screens, what you can do is you can move these to your advantage. So let's first talk about what each of these screens are. The Review and Edit is really your broader summary window. Again, it's giving you the study name. This is the one that's going to allow you to move between slides and wells. You can just toggle next well and go back previous well. It's also going to allow you to mark, once you're done editing a well, you can mark the review status here. So if you hit Mark Well, this would go to complete. And it's just a marker so that you know what the review status is. So if you have hit, let's say, slide well APIS, and we're in well 1, the numeric results in this window correlate to what's being shown in the Review and Edit window. And this Review Comet window is actually showing these individual comets. So in the Review and Edit, this is highlighted green. It's showing up in this is the first cell that's shown here. And it shows your tail moment, moment arm, percent DNA, the normal damage parameters. This window is actually gives you a broad overview of each of these comets. So this is the comp where this comet is positioned in the search image window. So if you are going to edit, I strongly recommend first starting with the search image window. This is the search image window. All of these are scalable, so you can always expand it to full screen if you want, or you can put it right back down to size. For the CometChip, it's nice to sort of scan down and make sure all the comets that were selected is still in the array panel, is within the micropore. So all these look pretty good. So that's the first step. But what you can do in these search images, suppose this one highlighted in white was actually not within the pore. You can right-click on it and it's going to exclude it. It'll exclude it here in the search image window. It'll show it excluded in the review window and also in your numeric results. And you can toggle back to unclick it. Okay, so like I mentioned, this is very nice because you can eliminate artifacts or hedgehogs within the search window. It's very fast. The second way of editing is you can go over here into the review window. I first recommend that you go under Edit and select Show Intensity Profile. So this is going to show the intensity profile relative to the comet. So in this first slide must have been untreated cells. So anyway, you can scan through here and check how well the markers. You have head markers. The white places the marker at the start, the red at the end of the head, or beginning of the tail, and the green is at the end of the tail. So anyway, you can go through here and look at where these markers are actually positioned. Let's say you wanted to move the cursors around. What you would do is in the review and edit, double click, and you have another window that comes in specific for this. It's only in this Edit Comet window, when you double left clicked, that this Edit Comet window is going to appear. So within here, I strongly recommend you go under File and you show the intensity profile before you begin moving these cursors. You also have this display range, so you have a high level, or you can click down to a lower level. And you can click on the marker to see exactly where it's been positioned. So again, the only markers you can move when editing the comet is for the start of the tail and the end of the tail. You cannot move this first position marker. So we'll get out of here. So like I mentioned, everything is pretty much a toggle. So let's go look at, let's see if we can find some where the comets weren't in the grid. Oh, here's a good one. You're looking for a panel. So right here, this comet right here was not within the pore. So again, this would be one that I would automatically exclude. And so you can see it's excluded here. And again, you can go through here and just sort of scroll down as you review to see whether or not you like how the comets were being called. Now, if you look at this one, this is small. You might question it. So let's click on it, left click, and see where it appears on the search image. So here, yes, it did fall within one of the micropores, so I'm not going to delete it. We have a comet here that looks strange. We have some type of strange tail, so let's click on this one. See, I really couldn't see it from the search image window, but I can see it in the review comet, so I'm going to delete that. So I would just right click. And let's look at one. Maybe I question whether or not how it called the tail. Let's see. These look pretty good, but let's go ahead and select this. We want to if I want to move the markers, you cannot move the markers in the review comet. So let's left click twice, go under File. So I already have the intensity profile showing from the previous image when I selected it. And again, I strongly suggest before you start moving these markers, you really look at where, look at in high exposure and low. So you can, if you want, go ahead and move it, but you'll probably find that it really doesn't affect the overall calculations by much. Okay, so let's click out. So like I said, you could have, as you're reviewing each one individually, you could have, say we're slide 1, I could have marked that well as review, so it was already completed. This is a marker for you. It's going to allow you to come in and out of the review and know where your position was previously. So let's assume we've edited everything. You can mark all wells. Once you mark all wells, the wizard is going to come back up and he's going to ask, do you want to print or export the study results now? So let's say yes. Now all the windows have closed. And now what we're seeing is the Generate Report window. In this window, it really depends. You have the software takes certain measurements, moment arm, moment of inertia, fragmentation, percent DNA in the tail, length, area, and intensity of comet. You also have a series of options of how you'd like to export. So let's say first of all, I only want to export the measurement percent DNA in the tail. And for my data options, I want all the raw data. I want to know what the protocol setting was that allowed me to draw the boxes around my image. And I also want the summary statistics. So you've selected all this. You can print it, but probably most people are simply going to want to export this data to Excel. So let's hit export. So it says the data is now on the clipboard and is available for pasting into another application. It's also asking you the question if you want to start a new study. But for right now, we need to go and we need to paste our data into Excel. So open up Excel. Okay, and we are going to paste our data. So when you paste the data, what you're actually seeing, this is the same data that you would see on a printout. So you have the study name, which we called 4X CometChip test images. The protocol, which was used to draw the boxes, was the default protocol that comes with the software. It's 4X CometChip alkaline comet. It was named this way because the images I typically take with CometChip are 4X. These are the protocol definitions that were used to generate this protocol. We're going to discuss how to generate a new protocol later in the presentation. Again, we had 3 slides in our study, and we had 2 well slides. And then following this is the summary statistics. We chose to export percent DNA in the tail. So the summary statistics, you have it for each slide and each well. It's going to show the number of comets initially found by the scanning software. The number counted is the number after you have completed editing, because you may have excluded some of the cells. So then for the summary statistics, since we only selected percent DNA in the tail, you'll get the mean, the standard deviation, the minimum, the maximum, and the median for that well. Underneath, we also selected we wanted to export the raw data. So below here you have so this is slide 1, well 1, and you identified so many comets which were arbitrarily numbered 1 through 171. So for comet 1, the associated damage parameter was 2.9% D in the tail. And if you scroll on down, you have it for all the, so here is well 2 on slide 1, etc. Okay, so let's now go back and let's say we want to now export our data in order to do a box plot. So you can select percent DNA in the tail and box plot. Now we're going to export. Whoop, sorry, got it. All right, percent DNA in tail box plot. Now we're going to export. Again, the data is on the clipboard, so we want to paste it. So let's just pick another sheet here. All right, so the data is exported in 2 columns. You'll have the study name, you'll have the sample name, which is basically the slide number and the well number. So these are 2 well slides. So we had the first slide 01 and well position A1. And remember, we had like 171 comets. So these were originally comets, say 1 through 171, but they're all being called off that same well number. But you still have the associated damage parameter. So you now have all the all the data listed. So you can also see here I've got slide 1, well 2. So in-house we use the Analyse It software. It's a plug-in for Excel. So let's go ahead and open Analyse It. We want to identify the data set, so we put our cursor within the data set, select data set. Okay, so over here the data set's 4x CometChip images. It selected all the data. These are the variables, so we're going to go ahead and say apply. So it's just recognizing the data. Then we're going to compare groups. Let's select median and quartiles. All right, then we have to start filling in information in order to make the box plot. So the Y response is going to be percent DNA in the tail. The X factor, that's your sample name. So we're going to click on mean, so we get the central moments based on pooled standard deviation. We want median and quartiles. We're going to do a confidence interval of 80. We're not going to do a box plot, a dot plot. We're only going to do a box plot, and we are going to select skeletal with a confidence interval of 80, and then just select calculate. And then we've automatically created our box plot with our exported data for these 6 slides. As you can tell, I had duplicate slides for untreated, and this is a low level of treatment, and then a higher level of treatment. And then Excel also generates, it'll give you the means, the medians for each of those wells, as well as the standard deviation or standard error. Okay, so now what we're going to do, it's important that, let's talk about the heart of the software is you have to be able to draw the boxes around the comets within your images. So let's go to protocol definitions. If you have an image, the first thing I would recommend is test some of the default protocols on an image. So the software allows you to do this. So let's first, like I said, my images were CometChip, taking it 4x magnification. So let's first just see how we test. So let's select a protocol called 4x CometChip alkaline comet. Go ahead and select OK. Now this protocol definition window comes up, and the box width and height have already been set. So what we're going to do is let's just test a protocol. So we're going to, or test an image to see if this protocol definition works well with our image. So let's hit test. Okay. So when you're first testing an image, I recommend starting with an image that has some damage on it, because you want to make sure that tail is encompassed within that box. So again, you're simply using Explorer to call up these images. So let's open the file. And there you go. You just tested an image. You can see that it was able to identify 167 comets using this default protocol. And what you're looking for is the white box being drawn around the comet. You need to know how many comets it could find with the protocol. And you don't want things being obviously left out. All right, so let's do an example where let's test a protocol. You can also get to these protocol definitions with the wizard. So let's go back. Now, my test image is at 4x, but we also have some default protocols suitable for 10x. So let's try the 10x alkaline comet protocol and see what happens. Well, this box width and box height are much larger for this protocol. But let's go ahead and let's test our image again. Again, take an image that has some damage in your comets. Let's open up your image file. Wow. Now you're not seeing white boxes around these comets. What you're seeing is a lot of overlapping blue. So this protocol that was used didn't work, all right? And sometimes maybe you got even 50% of the comets being called in your image, but you'd like much more. So this is an indication that you need to develop a new protocol. You need to create a new protocol. So how are we going to do it? All right, so To create a new protocol, what you're going to do is you start from a default protocol, right? So the default protocol already has some parameters built in. So the first step you have to do in creating a new protocol, open the default protocol, any one of them. The first thing you need to do is you need to measure the comet head on your control image. So you want a control image with no damage. You just want to measure the size of your comet head. So let's click Measure Head. Then what you need to do, step 1, select a representative negative control image. So click on the button to select an image. So let's select an image. So I know that my first slide 1, well 1 is my control. So let's select, open the image. Now step 2, to identify a cell, click on the center of the cell, press the button below to start the process. Okay, so hit select cell. The image comes up. Simply move your cursor, and I'd recommend expanding on this image. So find an untreated cell and click on the center. Okay, so it measured it. So step 3, click the button below to open the Comet editor so that we can now position to measure. So step 3, click on Measure Comet Head. And now you have another window coming up which allows you to have cursors. So move your cursor to the edge of the comet head and to the end of the comet head. Now select Done. All right. So the software is going to automatically perform some calculations. Just select Finish. Now the wizard comes up. Do you want to test these changes now on the control image? So say yes. All right. So now we have found a total of 167 comets. So this is really good. But you can notice that the boxes are a little large and you're probably, losing some data. So you can decrease the size of the boxes. So, okay. So now let's go back to this protocol definition. So all the changes are now here. So why don't let's decrease the comet width from 144 to 115. So I think I'm decreasing it by, 20%. Let's also decrease the box height, go from 83 to 66. All right, now let's test. And again, let's select an image. So again, let's test with the damage. All right, so click Open, and there you go. So now we've just identified one more comet instead of 167, 168, but you can see that the box sizes, they're no longer overlapping. So we're going to say OK. Right, so we're not done yet. So see, we still have this name called 10X alkaline comet. This is a default protocol that you're working on. So now you have to hit Save As to save your new protocol. So let's call this protocol Test. And then we are going to create and select OK. All right. So now what you can do is you can go back under these protocol definitions. You now have a new protocol called Test, which you can use to run your images. You can also, let's look at some of these default protocols. I want you to notice that you can never, ever delete. This is a default protocol. It's a built-in. It has a check mark. And so you can never delete it, and you can never overwrite it because these are marked out. So it is only with your custom protocols, see, that you're able to it's not built in, so there's no check, and you have the ability to delete, write over with the same name, or you can rename it. Let's see, so we can go ahead, we can delete this. Delete protocol definition, yes. One important thing that I'd like to note is, so when you create the study folders, which is basically your specific experiment that you're saving your images to, the data also for each comet is being saved to that folder. These folders can actually start to get fairly large. They're actually stored on your C drive, so on your computer. So if you select C, there's a Comet folder here, which the software always looks for. Within Comet, there is a Studies folder, again, which the software always looks for. But it's within these study folders that you have all your different individual experiments. So if you delete these, it's gone, and all your images are gone that you actually put in there. So one thing is always keep your images in a separate location in case you decide to delete your specific experiment. Or when these folders start to take up a lot of room, you can move the study folders into a different location. And if you need them, you can just move your experimental study back into the studies folder, which is in the Comet folder. So this concludes our demonstration of Comet Analysis Software. Thank you for your attention.